Picture this. You are standing in a museum looking at a reconstruction of an early Homo sapiens. The figure is posed dramatically, spear in hand, staring into the middle distance with the expression of someone who has just spotted something either edible or dangerous. The placard next to it mentions brain volume, tool use, diet. It does not mention skin color. You have probably never thought about why it does not mention skin color. You have probably never thought about skin color in this context at all. But here's the question that is sitting right underneath the surface of that museum display, quietly waiting for someone to ask it. What color was that person? And the deeper follow-up, why? Because the answer is not what most people expect. It is not ambiguous. It is not lost to time. The science on this is remarkably clear, and it tells a story about human evolution, biology, geography, and the way the human body responds to its environment that is one of the most elegant and underappreciated chapters in the entire history of our species. All early modern humans were dark-skinned. Not some of them, all of them. The full range of skin tones visible across the human population today did not exist at the origin of our species. It developed later in specific populations in response to specific environmental pressures through mechanisms that geneticists have now mapped in considerable detail. The diversity of human skin color that you see in the world today is young. The original human skin tone is old, and it is dark, and understanding why requires going back to a time before modern humans existed at all. Start with what skin actually does, because this is the foundation of everything that follows, and it is more complicated than most people appreciate. Skin is the largest organ in the human body. It is the interface between your internal biology and the external world, and it serves multiple simultaneous functions: protection from pathogens, regulation of body temperature, synthesis of vitamin D, and, critically for this discussion, management of ultraviolet radiation from the sun. Ultraviolet radiation, specifically the UVB component of sunlight, does two things to human biology that are in direct tension with each other. On one hand, UVB is necessary for the synthesis of vitamin D in the skin. Vitamin D is essential for calcium absorption, bone development, immune function, and a range of other physiological processes. Without adequate vitamin D, humans develop serious health problems. This is not a minor nutritional issue. Severe vitamin D deficiency causes rickets in children, bone softening in adults, compromised immune function, and a range of other conditions that significantly reduce survival and reproductive success. On the other hand, UVB damages DNA. Specifically, it causes mutations in skin cell DNA that lead to skin cancer, and, more acutely relevant from an evolutionary standpoint, it breaks down folate. Folate, also called folic acid or vitamin B9, is a nutrient that is absolutely critical for DNA synthesis and cell division. Without adequate folate, cell replication goes wrong. The consequences for reproduction are severe. Folate deficiency during pregnancy causes neural tube defects in developing fetuses, miscarriage, and developmental abnormalities. In adults, it impairs the production of sperm. An organism with chronically depleted folate has substantially reduced reproductive success, which in evolutionary terms is the same as saying it has reduced fitness. Here is the conflict. The same sunlight that you need to make vitamin D is also destroying the folate that you need to reproduce successfully. Your skin is caught between two essential biological requirements that are satisfied by opposite responses to sunlight. Melanin is the solution to this conflict. Melanin is the pigment that determines skin, hair, and eye color across humans and many other animals. It is produced by cells called melanocytes, which are distributed throughout the skin, and it exists in two primary forms: eumelanin, which is dark brown to black, and pheomelanin, which is reddish to yellow. The ratio and total amount of these pigment types determine the color of your skin. Melanin absorbs and scatters ultraviolet radiation before it can penetrate into deeper layers of skin where it would cause the most damage. Specifically, it protects the DNA in skin cells from UV-induced mutation, and it protects folate from UV-induced degradation. More melanin means more UV protection, means better folate preservation, means better reproductive outcomes in high UV environments. The anthropologist Nina Jablonski and her colleague George Chaplin did extensive research mapping the relationship between UV radiation levels at different latitudes and the skin color of indigenous human populations who had lived in those regions for long periods. The correlation is remarkably tight. Where UV radiation is intense year-round, near the equator, at high altitude, indigenous populations have the darkest skin tones. Where UV radiation is weaker, at higher latitudes, in regions with long dark winters, indigenous populations have lighter skin tones. The skin color map of humanity, before the mixing of populations through migration and trade, tracked the UV radiation map of the planet with a precision that is the clearest possible evidence that this relationship is not coincidental. It is selection. Evolution working on human skin exactly as it works on any other trait. The individuals with the most reproductively advantageous version of the trait in their specific environment produce more surviving offspring. And over generations, the population shifts toward that version. Now go back to the beginning of our species, roughly 300,000 years ago in Africa, to understand why darkness was the ancestral condition. Modern Homo sapiens emerged in Africa. This is not disputed. The genetic, fossil, and archaeological evidence all point to an African origin for our species, with the most ancient confirmed Homo sapiens fossils found in Morocco and Ethiopia. The populations from which all living humans descend were living in sub-Saharan Africa, near the equator, under some of the most intense ultraviolet radiation on the planet. But there is an important prior chapter here, because our ancestors were not always dark-skinned. Go back further before Homo sapiens to the common ancestor we share with chimpanzees and other great apes, and you find something interesting. Chimpanzees, our closest living relatives, have pale skin under their fur. Most great apes have pale to medium skin tones. The fur provides their UV protection. Skin melanin is not their primary defense against ultraviolet radiation. Early hominins, our ancient ancestors before they became fully human, were similarly covered in body hair that provided a degree of UV protection. But as the hominin lineage evolved, something critical changed: body hair reduction. The leading explanation for why humans lost most of their body hair is thermoregulation. As hominins began occupying open savanna environments and engaging in more sustained physical activity, pursuing prey, traveling longer distances, occupying more open terrain, the ability to dissipate heat became critically important. Dense body hair traps heat. Sparse body hair combined with a high density of sweat glands allows the evaporative cooling that makes sustained activity in hot environments possible without overheating. When body hair was reduced, the UV protective function it provided disappeared. Bare skin in equatorial Africa is exposed to intense ultraviolet radiation with no buffer. The folate-depleting and DNA-damaging effects of that radiation became immediate selection pressures on skin color. And, in that environment, dark, melanin-rich skin was selected for strongly and relatively rapidly by evolutionary standards. Genetic analysis supports this timeline. Researchers studying the MC1R gene, which is one of the primary genes controlling melanin production in human skin, have found patterns consistent with strong selection for dark skin occurring roughly 1.2 million years ago in the hominin lineage, corresponding approximately to the period when significant body hair reduction is thought to have occurred. The timeline connects. Hair loss created the selection pressure, and dark skin was the response. For most of our species' history, then, the default human skin tone was dark. The variation you see across human populations today is the result of subsequent migrations and adaptations that occurred after groups of Homo sapiens left Africa and spread across the globe. The question of when and how skin lightening evolved in non-equatorial populations is where the genetics become particularly rich and informative. The simplest framing of the pressure that drove skin lightening is the vitamin D argument. As human populations migrated to higher latitudes where UV radiation is weaker, particularly during winter months, the selection pressure that favored dark skin, folate protection, was reduced. While a different pressure, the need for sufficient UV penetration to enable vitamin D synthesis, became more important. Lighter skin allows more UVB through to the skin cells where vitamin D synthesis occurs, which is an advantage when UV radiation is scarce. This is the basic framework, and it is well supported. But the genetics tell a more complicated and more interesting story than a simple linear progression from dark equatorial skin to light polar skin. First, the timing. Genetic studies have found that the variants associated with the lightest skin tones in European populations are surprisingly recent. The most significant variant, affecting a gene called SLC24A5, which has a dramatic effect on melanin production, became common in European populations only within the last 6,000 to 12,000 years, which is the Holocene after the last ice age, during the period when agriculture was developing and spreading. This is unexpected if the simple vitamin D story were the whole explanation. Europeans have been living at high latitudes for far longer than 12,000 years. Why did skin lightening variants become so common so recently? Several explanations have been proposed. One focuses on diet. Hunter-gatherers who consumed significant quantities of animal products, particularly fish and organ meats, had dietary sources of vitamin D that partially compensated for reduced UV synthesis. The shift to agriculture, with its grain-heavy diet lower in vitamin D-containing foods, may have increased selection pressure for lighter skin capable of producing vitamin D more efficiently from weaker sunlight. The timing of skin lightening in European populations aligns roughly with the spread of agriculture, which supports this hypothesis. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Another explanation involves sexual selection. Some researchers have proposed that lighter skin, once it appeared, may have been preferentially selected through mate choice in some populations, accelerating its spread beyond what UV vitamin D pressures alone would explain. This is harder to demonstrate definitively and remains more contested. But it is not implausible given what we know about the role of visual signals in human mate selection broadly. The reality is probably a combination of factors, with UV vitamin D balance providing the foundational selection pressure and other factors influencing the specific timing and trajectory of change in particular populations. The geographic spread of human skin color diversity maps onto migration history in ways that are illuminating. East Asian populations who inhabit a broad range of latitudes show skin color variation that is generally intermediate to lighter, but the specific genetic variants responsible for their lighter skin tones are largely different from those responsible for lighter skin in European populations. This means that skin lightening in East Asian and European populations evolved independently through different genetic pathways in response to similar environmental pressures. This is a textbook example of convergent evolution, the same functional outcome achieved by different genetic means in different populations. Indigenous populations of the Americas who descended from ancestors who crossed from Northeast Asia into North America roughly 15,000 to 20,000 years ago and then spread rapidly across 2 continents, show skin tone variation that spans a wide range but does not, on average, lighten to the same extent as indigenous European populations at similar latitudes. One hypothesis for this is time. The Americas were populated relatively recently in evolutionary terms, and there has been less time for selection to drive skin lightening to the same degree seen in populations that have been at high latitudes for much longer. Another factor may be diet. Many indigenous American populations at high latitudes had diets rich in vitamin D from marine and terrestrial animal sources, reducing the selection pressure for lighter skin. Indigenous populations near the equator in Asia, Africa, and Australia independently converged on dark skin tones because they are all under the same intense equatorial UV radiation. Australian Aboriginal peoples, who descended from ancestors who arrived in Australia around 50,000 years ago, and whose equatorial forebears were dark-skinned, maintained dark skin tones in tropical and subtropical parts of Australia where UV radiation is intense. The convergence of dark skin tone across geographically separated populations in high UV environments, achieved through partially overlapping genetic pathways, is compelling additional evidence that melanin concentration is being directly selected for by the UV environment. There is a specific gene that deserves extended attention because its story illustrates the evolutionary dynamics of skin color more vividly than almost anything else in the literature. The MC1R gene, mentioned earlier in the context of early selection for dark skin, encodes the melanocortin 1 receptor, which plays a central role in determining the ratio of eumelanin to pheomelanin produced by melanocytes. In most of the world, the functional version of this gene produces predominantly dark eumelanin, resulting in dark brown to black skin and hair. Variants of this gene that shift production toward lighter, redder pheomelanin are associated with the lighter, often freckled skin, and red or auburn hair seen in some European populations, particularly in Ireland, Scotland, and other parts of northwestern Europe. What makes MC1R fascinating from an evolutionary standpoint is its behavior in African populations. In sub-Saharan African populations, MC1R shows very low genetic variation. Almost everyone has the same functional version of the gene, the one that produces dark skin. This is called a selective sweep. The evolutionary pressure for dark skin in this environment is so strong that variants that reduce melanin production are rapidly eliminated from the population because individuals carrying them have lower reproductive success. The gene does not have room to vary in equatorial Africa because any variation away from maximum melanin production is immediately selected against. Outside Africa, in populations that have been living at higher latitudes for many generations, MC1R shows much higher variation. The selective constraint has been relaxed, not because dark skin became actively harmful, but because the intensity of the selection pressure for maximum melanin decreased enough that variants could persist in the population and eventually increase in frequency if they provided vitamin D advantages. This asymmetry in genetic variation, constrained in Africa, diverse outside Africa, is itself strong evidence for the functional role of MC1R in human skin color evolution and for the directionality of the story. Dark was ancestral, and strong selection maintained it where it mattered most, while relaxed selection and new pressures drove diversification elsewhere. Address a misconception that comes up regularly in popular discussions of this topic. The idea that skin color variation across human populations reflects something fundamental and ancient about those populations, or that different skin colors represent different branches of humanity with deep, separate origins. The genetics of skin color do not support this interpretation at all. The genes primarily responsible for skin color variation across human populations are a small subset of our total genome, and they show patterns of variation that are recent relative to other aspects of human genetic diversity. All human populations share the overwhelming majority of their genetic variation. The genetic differences between any two human populations are smaller than the genetic differences that exist within those populations. Skin color is a surface adaptation. It is the body's response to the UV environment at a specific latitude. It evolved multiple times independently through different genes in different populations, in response to the same environmental pressures. Two people with identical skin tones may have achieved that skin tone through completely different genetic pathways if their ancestors adapted to similar UV environments by different routes. Two people with very different skin tones may be more closely related to each other in terms of total genome similarity than either is to a third person with an intermediate skin tone. Skin color, in other words, is a very poor proxy for genetic ancestry in the deeper sense. It is one of the most visible human traits and one of the least informative about overall genetic relatedness. Evolution made it highly variable because UV radiation varies dramatically with latitude, and that variation matters for biology. But making skin color variable for that functional reason required almost no change to the vast majority of the human genome. Here is something that surprises most people when they first encounter it. The study of ancient DNA, which has exploded in the last decade as sequencing technology has become powerful enough to extract and read genetic material from ancient skeletal remains, is directly confirming the recency of light skin in populations where it is now common. Ancient DNA from European populations of the Mesolithic period, roughly 12,000 to 6,000 years ago, shows that many individuals carried genetic variants associated with darker skin tones and blue eyes, a combination that is now rare and that illustrates how specific traits can shift independently through selection. These were fully modern Homo sapiens, anatomically identical to contemporary Europeans, carrying skin color genetics that would have corresponded to considerably darker skin than is typical in modern European populations. The transition to lighter skin in European populations appears in the ancient DNA record as a relatively rapid change in the frequencies of specific variants during the Neolithic period, corresponding to the arrival and spread of agricultural populations and the changes in diet and UV exposure that accompanied the shift from mobile hunter-gatherer life to sedentary farming. This is direct observational evidence, not inference. Ancient DNA is telling us, explicitly, what skin color variants existed in specific populations at specific times. And it is confirming that the range of skin tones visible in human populations today is the product of recent evolution, shaped by migration, diet, and UV environment layered on top of a deep ancestral baseline of dark, melanin-rich skin. The practical implications of this evolutionary history show up in modern medicine in ways that are worth understanding. Vitamin D deficiency is more common in darker-skinned individuals living at high latitudes than in lighter-skinned individuals in the same environment. This is not a dietary or behavioral difference primarily. It is the biology working exactly as it was designed to. Dark skin that evolved to protect against intense UV radiation in equatorial Africa provides more UV blocking than is beneficial in a low UV northern environment. The same adaptation that was protective in one context creates a nutritional vulnerability in another. This has real health consequences: higher rates of vitamin D deficiency-related conditions, including bone health issues, immune function impacts, and research suggesting connections to a range of other health outcomes in dark-skinned populations in high-latitude countries. The solution is not complicated. Dietary vitamin D supplementation and food fortification compensate for reduced UV synthesis effectively. But recognizing that the vulnerability exists requires understanding the evolutionary context. Conversely, very light-skinned individuals in high UV environments face elevated skin cancer risk because their melanin levels provide less protection against UV-induced DNA damage. Melanoma rates in light-skinned populations in high UV environments like Australia are among the highest in the world, reflecting the mismatch between skin tone adapted to low UV European latitudes and the intense UV radiation of the Australian continent. Both of these health dynamics are legible only through the evolutionary lens. They make no sense in isolation. In the context of what skin color is, an evolved calibration of UV protection versus vitamin D synthesis. They are exactly what you would predict. Step back and look at what this entire story means, because it is worth taking a moment to appreciate how profound it is. Every human alive today descends from dark-skinned ancestors. The skin color diversity that exists in the contemporary human population, every tone, every shade, from the deepest brown to the palest pink, differentiated from that single ancestral starting point within the last roughly 100,000 years, and in some populations much more recently than that. In evolutionary time, that is the blink of an eye. The mechanism that created this diversity is not mysterious or complex. It is one of the most elegantly simple examples of natural selection in the human body. The sun is intense near the equator and weak near the poles. Folate and vitamin D have opposite requirements for UV exposure, and melanin is the molecule that calibrates the balance between them. Evolution adjusted melanin levels in human populations that moved to new UV environments, and the result is the range of human skin tones. Race, as a biological category built on skin color, collapses under the weight of this knowledge. Not because skin color variation does not exist, it obviously does, but because it is too superficial, too recent, and too functionally specific to carry the weight that has been placed on it throughout history. The genes responsible for skin color variation represent a tiny fraction of our genome. The deeper genetic story of human diversity is one of shared ancestry. recent divergence, and the overlay of specific adaptations to specific environments on a foundation that all humans hold in common. The person in that museum display with the spear and the dramatic expression was dark skinned. Every Homo sapiens who came before a certain point in our evolutionary history was dark skinned. The diversity that followed is one of the most rapid and visible examples of evolutionary adaptation in our species, driven by something as simple and powerful as the angle of sunlight hitting the Earth at different latitudes. That is not a minor footnote in human history. It is the opening chapter, and understanding it properly changes how you see every face you have ever looked at,